Hi, I'm Bella,
I'm Anthony.
and we're presenting on the characterization of Topoisomerase II in human cells. What we see to the left over there is a metaphase human cell rotating through, and it's showing all the different channels. All you can see there is just the DNA cloud.
What are homologous chromosomes? First off, I want to talk about what chromosomes are. Chromosomes are that contain genetic material such as DNA. This DNA is then processed throughout the cell. Homologous chromosomes are of chromosomes that has genetic material from the father, paternity derived, and from the mother, maternity derived. And as we see here in the figures, we see a centromere on the homologous chromosomes. And I'm going to talk about segregation of homologous chromosomes. Homologous chromosomes segregate to opposite sides of the cell. Segregation refers to how genetic material such as DNA is being sorted and distributed throughout the cell. And the mechanism of segregation that we want to figure out is unknown.
Since the mechanism is still unknown, we've developed a working model that there could be physical linkages between each haploid set of chromosomes, keeping each pair of them not being on the same side of the cell. We think that these linkages could be made up of DNA, so we're staining for Topo2, which is used in organizing DNA.
Significance understanding the mechanism of segregation of homologous chromosomes and might provide avenues toward chromosome abnormalities. Our hypothesis is how topoisomerase II may be present on these linkages and may play a role in the segregation of homologous chromosomes.
Our experimental approach is mostly in confocal microscopy. The image to the right is an image of a confocal microscope. The confocal microscope uses lasers to excite the cells, and then we can see on the left here what it actually looks like for us when we're looking at the confocal. All of the different colors are separate channels, and they're separate lasers that the confocal uses to picture the different proteins. The red is the centrosomes, and we see those completely on their own. The blue is the DNA stain, and then the green is the Topo2 stain. The last image is what we can see once we put it into a computer program called Imaris to visualize all the channels together.
All right, so what we see here is topoisomerase II at metaphase specifically, its distribution. So we can see these three figures being present with all three channels. And then we have the other figures that present only the protein with the centrosomes and the other one with DNA and the centrosomes. As we can see in the first figure, we see how all three channels are being distributed and how topoisomerase II may play a role in the haploid set segregation, since we see this low centromeric staining along the center of the centrosome axis. We can see it. And also in the other figure, we see topoisomerase II with the centrosomes, how it's very distributed and very condensed. How we see how it's distributed throughout the cell, and then the DNA, just the DNA mass we see with the centrosome, just the chromosomes. Yeah.
So once we gather all of those images that we've seen on the last slide, but for a bunch of different cells, we go ahead and we'll make heat maps so we can actually see if the trend we saw in the last cell is present throughout all of the cells that we have images of. To do this, we make heat maps. So the first step is to divide them up into just the DNA stain and just the Topo2 stain. The next step is to align all of them by the centrosomes, which are those red dots on either side of them. And then we put them into a software called ImageJ to make heat maps that we can see over at the side. The blue means that there's not very much overlap between the cells, and the bright white means that there's a lot of overlap between the cells. Okay, so we see with the heat maps that there is a low region for our Topo2. We can see that it's along the centrosome axis. And we don't see this at all in the DNA stain, which is a good sign because that means that Topo2 has a different distribution pattern than the general DNA does. For conclusions, this means that Topo2 may be involved in chromosome set segregation, which means that we're one step closer to figuring out what the mechanisms are that could be segregating these sets of chromosomes. For future directions, we hope to stain the centromeres with the Topo2 so that we can see if the Topo2 is between the centromeres or not. And we also would like to do some genetic perturbations of Topo2 to see if the haploid set segregation stays around when we do remove some of the Topo2. acknowledgements, thank you to all the members of the Hua Lab and Dr. Hua herself, and to our graduate mentor, Omar, for helping us on our project.